Hi dear students, a warm good morning to all of you and welcome to the microbiological examination of water. Before I move on to the microbiological examination of water, which microbes do we have to identify within water? That is of very much importance. We know that it is not practical always to find out all the pathogens which have been present within the water because pathogens are of a wide variety. So we choose to find out indicator organisms within the water. And what are these indicator organisms? These are indicator organisms which could be giving an indication for us that probably a pathogen is also being present within the water. Because if we go to try to find out each and every pathogen from water, uh, it is being found to be not economical in a nature. So we choose to find out indicator organisms in the body, in the water, and these indicator organisms should be indicating that probably a pathogen is present. Now there are some conditions which we choose for the indicator organisms. Sometimes these are organisms which should be useful for all types of waters and they should be present whenever the enteric pathogen is present, and the organism should have a reasonably longer survival time than the hardiest enteric pathogen, and the organism should not grow in water, and the testing method which we use to find out this indicator organism should be easy to perform, and the density of the indicator organism should have a direct relationship to the degree of fecal pollution. And the organism should be a member of the intestinal microflora of warm-blooded animals. Now, you might have noticed here, like we're talking about enteric pathogens. So, enteric is coming from the gut. So, when we are trying to find out enteric pathogens from the water, we try to find out indicator organisms which are resident in the gut of the animals. So, if the water which we examine happens to contain a enteric organism within the water which we analyze, it will indicate that it has a fecal, the water has a chance of fecal pollution. So I guess you got an idea what is a indicator organism. And the estimated level of indicator organisms will vary in different things. Now, if you go to consider raw sewage, the coliform estimated level is 10 raised to 7 to 10 raised to 9. The fecal coliforms are 10 raised to 6 to 10 raised to 7. The fecal streptococci are 10 raised to 5 to 10 raised to 6. And you can also find coliphages, bacteroides, pseudomonas aeruginosa, staphylococcus, which is coagulase positive, etc. Now, if you go to consider the whole type of organisms which are being which are included under the group of Enterobacteriaceae, uh, the whole family of Enterobacteriaceae can include different things. It can include the coliforms, okay, as well as other members such as the Salmonella and Shigella. And among these total coliforms, we are in fact searching for the coliforms, which are the indicator organisms. And among these total coliforms, the fecal coliforms are a very good indicator organisms, which indicate that fecal pathogens are being present within the organism in the water. And among the fecal coliforms, we specifically check if Escherichia coli is being present within the water. So, the indicator organisms which we use most commonly come under the family Enterobacteriaceae, and they will be coliforms usually. And it's not that all Enterobacteriaceae are coliforms. We search for indicator organisms such as coliforms, and more specifically talking about fecal coliforms. And among fecal coliforms, the presence of E. coli indicates that the water is being contaminated by enteric pathogens. So, what do you mean by the total coliforms? The coliform group, it mainly includes Escherichia, Citrobacter, Enterobacter, and Klebsiella species, and they are relatively easy to detect. And if you go to specifically look onto this group, they include aerobic and facultatively anaerobic gram-negative non-spore forming rod-shaped bacteria, which is capable of producing gas upon lactose fermentation in a prescribed culture media within 48 hours at 35 degrees centigrade. So just let me just take it, explain it in an easy manner. If you want to find a coliform is having a property that it can utilize the sugar lactose, and after utilizing it within 48 hours at 35 degrees centigrade, this coliform usually will produce what? Gas. And coming to its features, how it looks, it is been found to be gram negative in nature. It is non-spore forming. You know what's a spore? A spore is something which has been produced by a bacteria when adverse condition comes. So the coliforms are non-spore forming and they are being found to be rod shaped. And regarding its growth, they can include aerobic as well as facultatively anaerobic forms. So if you find the coliforms, it is an indication, it is a good indicator organism, and it indicates that the chances of fecal pollution is there in the water. And the coliform group has been used as a standard for assessing fecal contamination of recreational and drinking water since early in the 20th century. And through practice, it has been learned that the absence of these organisms in 100 ml of drinking water ensures the prevention of bacterial waterborne outbreaks. But of course, sometimes some deficiencies also do exist. So if you find coliform within your water body, uh, it indicates that there is your water body of study is having a fecal contamination. And the deficiencies of, though we use coliform bacteria as indicators, of course sometimes some deficiencies also do occur in this test because regrowth happens in the aquatic environment, regrowth happens in the distribution systems, and sometimes suppression by high background bacterial growth could happen in coliforms, and it is not a indication of a health threat, and it does not have any relationship with enteric protozoans and viral concentrations. But however be the deficiencies, we still follow the detection of coliforms as a indicator organisms. And we should keep in mind that no one indicator meet all these criteria. And so process indicators are used to assess the efficacy of a treatment process. Like now, for example, and we will use also fecal indicators to indicate the presence of fecal contamination. And an index organism represents the presence and behavior of a pathogen in a given environment. So you keep in your mind to, instead of checking the pathogens in the water, you check for the indicator organisms, and among them the coliform are the one which we frequently use. And according to the US FDA, there are some standards for the water that which we use. Now, the Safe Drinking Water Act says that zero coliforms should be present in every 100 ml of the water which we analyze. And regarding the wastewater discharges, the permitted coliform is 200 fecal coliforms per 100 ml, and the sewage sludge should have less than 1000 fecal coliforms per 4 gram, and 3 less than 3 salmonella per 4 gram. These are there. So keep in your mind every regulatory body, especially in different countries, but there is a regulatory body which says that the coliform should be at this particular count for the safe drinking of the water, as well as for the sewage, as well as the wastewater discharges. And always zero coliforms per 100 ml is preferred for the safe drinking water, because that will indicate that what these fecal contaminants or fecal pathogens are not being present within the water which you drink. Now, as we all know, there are different methods which we have to detect various organisms. Either you can use the adsorption elution method, membrane filtration method, the cartridge filtration method, the cell culture method for the, or the cytopathic if you want to check the viruses. If you want to check the bacteria, you have a method where you grow it on the agar and then you count it. And if you want to check the parasites, you have the immunological staining methods and all that. So we, in all these methods, what are we trying to do? uh, you can either detect the concentration of it, or you can also count the bacteria, the virus, or the protozoa and all that. So let's try to find out a method where we what estimate the number of the coliform. And MPN, or the most probable number method, is mainly used to find out the number of coliforms which have been present in the water. So what we do is, the water to be tested is diluted serially and it is inoculated into lactose broth. And coliforms, if they have been present in the water, which we are checking, they will, as I told you, coliforms have the ability to what ferment lactose and produce acid and gas. So, uh, if you add an indicator into the solution, what happens? The color changes of the medium and the presence of gas is detected by when a Derams tube has been added. If gas production is happening, you can see that also. So if in a media, if you take the water and put it into a media containing lactose, and if a coliform or organism is present in the water, it will utilize the lactose present in it, releasing gas as well as changing the media color, and so that we can detect it. And using this, not only the presence of a coliform can be detected, you can also make a total number of coliforms which have been presented by counting the number of tubes giving a positive reaction and comparing the pattern of the positive results with a standard statistical table. So I will just help you in that. So the MPN test is having three types of tests. First, we have the presumptive test, which is a screening test to sample the water for the presence of coliform organisms. And if the presumptive test is negative, no further testing is performed and the water source is considered microbiologically safe. But if no further zinc is performed, the water source is considered to be microbiologically safe. But if you find that some tubes have what uh, tubes are positive with acid and gas, then we move forward with a confirmed test to make sure whether the what is a coliform present. And the method of presumptive test will vary for treated as well as untreated water. So depending upon the water which you are analyzing, the presumptive test will be different. Now consider if you are taking a untreated water. In the case of untreated water, what happens? You will take three set of test tubes. Okay, you are five of them will be having what? They will be of double strength broth, and five of them will be single strength, and five also will be single strength. And what you do is, in five of them, you will inoculate 10 ml of your sample, and five of the next one, you will inoculate 1 ml of your sample, and in the last one, in the last five, you will take zero point 0.1 ml of the sample, of the water sample, and then you plug the water and you plug it and you incubate it for 24 degrees centigrade. 24 hours, 24 hours plus 2 at 35 degrees centigrade. Now you might have a doubt, what is a double strength and what is a single strength. Just think, if I am saying you need to prepare what? A glass of Complan with 1 spoon of Complan in it. That is single strength. So here, when you prepare lactose broth with 1 spoon, I'm just telling you just in layman's language, okay? If you want to prepare a double strength, you means you take 2 spoons of that particular nutrient. So here, the nutrient or the broth which we prepare here is lactose broth, and compared to those two, what happens? It will contain double the amount of the particular nutrients compared to the single strength. So in the double strength broth, you take 10 ml of your sample. In single strength first set of tubes, you take 1 ml of water into each, and in the last one, you take 0.1 ml of the sample. And after that, you incubate them. And if it is positive, what would happen? acid production could be found, which could be determined by the change in the color. Okay, so you will add an indicator which will show that an acid has been formed or not. And then if gas has been formed, you will have what happens. This is called the Derham's tube. See, you can see a small tube within the test tube, that is called the Derham's tube. If gas is being formed, what happens? There will be a bubble formation, and uh, a negative presumptive, or the it immediately includes that the absence of gas will happen in the broth tube, and it will indicate that what it is negative. And this tube you have to keep it for 24 hours more to make sure that it is been found to be negative. And once you get the presence of gas or not, you can make, using an standard MPN chart, you can find out what is the number of coliforms which have been present. Now, among the first 5 tubes of 10 ml, if 2 of them are positive, and among the next one, 5 of 1 ml, 0 are positive, and in 5 of 0.1 ml sample added 1, it is 200, then the MPN per 100 ml is 5. And of course you have a 95% confidence limit, that is if it is, now if you're getting the 5 answer over here, maximum it can go up to 13. Okay, that's what it means. So like this you calculate what is the MPN value, or I'm telling you, to find the number of coliform test, you can use the MPN method, or the most probable number method, to find the what the count of the coliforms which could be present. Now, once you see the presumptive test, after that we can go to the confirmed test. Some organisms other than the coliforms also will produce acid and gas from lactose fermentation. And in order to confirm the presence of the coliform, you do the confirmatory test. So what do you do? From each fermentation tube which gives a positive result, you will take one loopful and put it on into a 3 ml lactose broth, or a brilliant green lactose fermentation broth. Uh, then you put it onto an agar slant and also to 3 ml krypton water. And then you incubate the lactose broth in tubes at 37 degrees centigrade, and you inspect for the gas fermentation for 24 or plus 2 hours. If no gas fermentation is seen, then you further incubate it for maximum of 48 hours to check for the gas production. And the agar slant should be incubated at 37 degrees centigrade, and gram stain preparations should be done from the slants to check whether it is positive or negative, because we know that the members of the coliform bacteria, they are gram negative non-spore forming bacilli. So on the agar, as you can just check whether the what they are showing the same characteristics. And the absence of gas formation in lactose broth, or the failure of demonstrating a gram-negative non-spore forming bacilli in the agar will show that it is not an indicator. It is not a coliform, but it is something else which was producing the what coliform, which was producing the acid and the gas. Then ultimately, we also have the completed test. Since most of the positive results from the confirmatory test may be false, it always desirable to do a completed test. And for this, what you do is, from the positive inoculum, you take bacteria and you take a loopful and streak it on a plate containing EMB, or the endo agar. And in this process, from every tube where you get a positive result, what you do is you take a loopful and streak it onto the eosin methylene blue agar, or the endo agar, and then you will incubate it at 37 degrees centigrade and one at 44.5 degrees centigrade. Now, why do you have to do this is, if the fecal contamination is from the warm blooded animals, you can indicate that by its growth in 45, and the other one is at 37 degrees centigrade. So from the confirmed test, when the gas is being present, you take some and streak it onto the EMB or the endo agar, and after that you check for how does the colony look like. Okay. Now, how can you infer whether it is a coliform test? It is a coliform bacteria. Now, if it is in EMB agar, if it's a positive reaction, you will form nucleated colonies on EMB agar. And other bacteria which will not form nucleated colonies. Now, what do you mean by nucleated colonies? Let me just explain. If you have a if you have a colony which has been found to be round in shape, at the center of that, somewhat similar to that of a nucleus of a cell, you will have a nucleation line. Like the presence of a nucleus, another particle would be present on the colony itself. So that will indicate that it is a coliform bacteria. And if you go to look into endoagar, lactose fermenting colonies and the surrounding media will appear to be as red in color if you are using a endoagar. And non-lactose fermenting colonies and the surrounding media will remain colorless for the for what for the what endoagar which we used. Now the same way you can do this for treated water also, but here you take one tube of single strength 50 ml and 5 tubes of double strength 10 ml for each water that has been sampled, and using a sterile pipette add 50 ml of water to the tube containing 50 ml single strength media and add 10 ml of water to 5 tubes containing 10 ml double strength media and incubate it at 37 to 45 37 degrees centigrade for 24 hours, and the process continues. And of course the MPN chart which you use here also would be different. For treated water and for untreated water we have different thing, and the MPN value of 16, that is the water sample contains an estimated of 16 coliforms per 100 ml if you take that. So for treated water as well as untreated water, you have different MPN charts. Now this is a treated water. For treated water, the MPN chart is like this. The sample volume also what we take, and the tube the same what we take is also being found to be different in the case of what treated and untreated water for the presumptive test. I hope you remember what is it for the what untreated water. In the untreated water we were taking like this. It is for untreated, you take, I told you about 5 test tubes with double string, 5 tubes with single string and 5 tubes with single string. But if you go to consider the what treated water, what the amount of test tubes and all that you take will be different. You will have a 1 tube of single strength with 50 ml and 5 tubes of double strength for each water with 10 ml. Okay, and the chart would also be different which you use for the MPN analysis. And the high temperature incubation is for the detection of the thermotolerant E. coli, and for following incubation the plates are being examined for the presence of typical colonies. And coliforms produce colonies with greenish metallic sheen which differentiates it from the non-coliform colonies. So the non-coliform colonies will not have a sheen. What do you mean by sheen? A sheen is something which like a shiny like appearance. Okay, a green metallic sheen will be found by the coliforms. And the presence of E. coli will have a greenish metallic sheen. Nucleated colonies will be present, and that indicates that E. coli is there. Now, the advantages of MPN test is the ease of interpretation, like either by observation or by gas emission you can find out, and sample toxins get diluted, and it's an effective method for analyzing highly turbid samples such as sediments, sludge, mud, etc. And it cannot be analyzed by membrane filtration, but the disadvantage is that it takes long time to get the results because you have to wait every 24 to 48 hours to give a proper result, because you have the the presumptive, the completed as well as the confirmed, and then the completed test to be done, and requires more hardware and glassware and media, and sometimes also we could get false positive results. So we also have other methods like membrane filtration for determining the coliform bacteria. What you do is you can take a filtration unit. Onto that, what is it, you keep the filtration paper and you pour the water onto it so that all the bacteria gets attached onto the membrane filter, and then you take the membrane filter and then you what place it onto the agar, Okay, and so after the process of vacuum filtration, you take the, and you just remember here the tube is connected to a vacuum, so the water gets comes out very fast, goes down into this when the vacuum is pump is being applied. Then you take this membrane filter and place it on the agar and find out the number of colonies. Now the other method is also of detecting indicator organism is a colloidal test, that is the addition of salts and enzyme substrates to the water sample, and if there is a yellow color, it indicates that what, the presence of coliform bacteria is there, and fluorescence under long wave ultraviolet light is also indicating the presence of E. coli. So these are some of the tests which we can have for the what, E. coli. Now what is the substrate which we added here? The substrate which we put onto that is orthonitrophenyl galactopyranoside, ONPG. Okay, ONPG is the substrate which we put into this water, and this is being, when this powder or ONPG, orthonitrophenyl uh, phenyl galactopyranoside, it will be broken down by galactosides, which has been present by coliform bacteria, and then you will get nitrophenol. And when you add a fluorogenic substrate, now for example, methyl amylophenol glucuronide or MUG, okay, that will produce a fluorescent end product after interaction with the enzyme glucuronidase found in the E. coli. And that is what which we are seeing here. This one, mug has been put, and after mug has been put, what happens? When you put the UV light, it will fluoresce, and thus we can indicate that E. coli is being present. So the Corley load test is performed by adding a small, taking the sample of a single bottle like presence or absence, or you can use MPN tubes that contain powdered ingredients consisting of salts or specific enzymes. And after 24 hours of incubation, you will check whether the coliform bacteria are being present by checking whether what, as we told, if it is containing, if it's converting into yellow color when you add ONPG, that is also positive colloidal test, or when you add mug into the water, if it fluoresces, that also indicates a coliform, a positive coliform test. So with this, we come to an end of the first session of the microbiological examination of water, and we were specifically talking on the what? The examination of coliform bacteria, the indicators of pathogenic organisms within the water. Thank you for now.